afternoon. I'm Beth Rosen Sheidley. I'm from Boston, Massachusetts. It's a pleasure to be here. I want to thank Rafaela and Francesca for inviting our group to speak. I work with Dr. Ann Paduri, who was one of the authors on the somatic mosaicism paper that was summarized so beautifully this morning. When we got the invitation to come, we thought, well, we're relatively newcomers to the field of PCDH19, and we don't really have research findings to share with you. On the other hand, we somewhat recently started an epilepsy genetics clinical service at Boston Children's, and we've diagnosed a number of children that we follow, and so we thought it would be helpful to at least share with you our experience, which I think will be very interesting, actually, to many of you, because the U.S. experience in terms of testing in particular, I think is quite different from what I've heard so far this morning that's taking place in European countries. So really what I wanted to cover this morning was a little bit, this afternoon, was a little bit of background about our clinical service, to describe the cohort of girls that we follow at Boston Children's, talk some about the psychosocial impact of testing. I'm a genetic counselor by training, so that is my area of expertise. So what I'm going to talk about is by no means a systematic study of counseling issues, but more anecdotally based, and then talk a little bit about future directions at Boston Children's and how we hope to collaborate with those of you who are here today. I really want to thank Dr. Legurn for setting me up beautifully to talk about genetic counseling issues. So, you know, Ricardo this morning, I often speak with my parents on the telephone, and like his mother, I think my parents really have no idea what it is that I do. I think my dad still thinks that I am involved in genetic cloning, and my mother looks at me and my little blonde-haired, blue-eyed children and says things like, well, I think they got all of their genes from your husband. They didn't get any genetic material from you. And so it's a good reminder to me of what people who are well-educated in the States, but perhaps not so well educated about science and medicine, might think about genetic issues. And it's always a good reminder to me when I have these conversations with them that I need to carry this into my work as a genetic counselor and never assume that the person that I'm sitting with has any particular understanding of genetic concepts or scientific concepts. So I put this definition of genetic counseling up here for those of you in the audience who may not be as familiar with the field that I've been part of for about 20 years. Really, my role is to help explain genetic test results to families, to help them adapt to that information, to provide them resources, both in the moment they're getting the diagnosis and over time. And to some extent, in the role that I'm involved with right now, part of what I do is to help to better understand the family history and to help facilitate decision-making around testing before that testing even takes place. The first exposure that I had to epilepsy in the genetic counseling setting was really as a student, so this is more than 20 years ago, when I sat in shadowing with a practicing genetic counselor. This is part of the training that takes place for genetic counseling students. And there was a young couple with an ongoing pregnancy, and the mom had epilepsy and was being treated with anti-epileptic medication. As it turned out, the dad also had a history of seizures and intellectual disability. And as this family history was elicited by the counselor, the dad became very emotional and began to cry. And when he was asked why he was crying, he said, well, because if there's something wrong with the baby, I know it's going to be my fault. And I will tell you that 20-something odd years later, now that I'm involved in epilepsy genetics, The counseling issues really haven't changed so much since then, even though our ability to make molecular diagnoses has changed quite a bit, as we'll talk about. So the genetic counseling profession is relatively young. There's been training programs in the US for master's level genetic counselors for about 30 years now. There are 35 or 40 training programs based in the United States and a few in Europe, particularly in England. I know there's at least one in Australia. There are several in Canada. And so it's usually a 2 to 3 year training process with equally balanced work in both the molecular biology and human genetics and the counseling aspects of the profession. Historically, genetic counselors have worked in prenatal settings like the one that I described, but also in pediatric settings with kids who have different types of disabilities or genetic conditions. Also, counselors, many of them work in cancer genetics or in specialty areas like neurogenetics. I myself started out in pediatric and prenatal genetic counseling, but most of my work over the last 20 years or so has been focused on autism, on psychiatric disorders, and then more recently, I've been involved with epilepsy. So I have to make a little joke about this picture because this banner across the front of the hospital where I work says that U.S. News and World Report ranks us as the number one children's hospital. In fact, we have slipped to number 2, so in case anybody from Children's Hospital Philly sees this, I wanted to point that out. Although apparently in neurology we are still ranked number 1, which is not important at all, but the neurologists at Children's Hospital like to joke about this, so I wanted to make a point of saying something about that. But the neurology department at Children's is rather large. There are 40 or 50 attendings in the department, and 11 or 12 of them are specifically epileptologists. There are a small number who are quite well-versed in genetics, including the 2 that I work with, and that's Dr. Paduri, who I mentioned, and Heather Olson, who is new to our group. So about 3 years ago, when I joined their team, we started an epilepsy genetics program, which is focused on providing clinical care to families. So we have a clinic that meets once a week, where we might see 2 or 3 patients every week, usually with early onset, difficult to treat epilepsy. So not always PCDH19 related. We've seen a lot of children with SCN1A mutations and Dravet syndrome, and quite a few other underlying genetic diagnoses. We also have a research arm to our program, so our main focus has been on Ohtahara syndrome and Dravet syndrome. And as I mentioned, we are somewhat newer to PCDH19, but in the last year or so, we have diagnosed quite a number of girls. So this has become something that's become more of interest to us in recent months. So in terms of the testing landscape in the US, something very dramatic has happened in the last year and a half. Up until then, we made diagnoses based on the clinical presentation. We would assess the medical history, the seizure history, and the family history, and make a decision about testing for a single gene based on that information. And there were a number of labs that offered testing specifically for PCDH19. But about a year and a half ago, GeneDx, which is a laboratory based in Maryland, launched what we call an epilepsy genetics panel test. So when they first began, they offered a panel of 30-something odd genes that were all related to different early onset epilepsy phenotypes. So essentially, it became less important for a clinician, let's say, who wasn't particularly knowledgeable to have a good understanding of what circumstances they might need to order a specific test, because they could order the panel and have everything looked at simultaneously. In part, this has become very appealing and widespread in the US because it is much more cost-effective. We have to deal with insurance issues in the States that I think many other countries don't necessarily have to think about as much. So some of our patients have private health insurance. Some of them have federal insurance or state health insurance. But these tests, if you start doing them sequentially one by one, become very expensive. They're several $1,000s a piece. And if you don't hit the right diagnosis the first time or the second time or the third time, you're talking about a bill that can reach into the tens of thousands very quickly, and it can be very time-consuming every time you order a test, waiting 6 or 8 weeks for those results to come back before you can order the next one. But that's the way that we used to do the testing before these panels became available. The panels are still relatively expensive. They tend to cost around $4,000 or $5,000, but you can get results back on anywhere from 70 to 300 different epilepsy-related genes within 6 or 8 or 12 weeks, depending on the lab that you choose. So these labs that I've represented up here are by no means the only ones that offer epilepsy genetic testing in the US, but they're the bigger players in terms of the large panel tests and the number of samples that they tend to get from providers. So this has been a big change, actually, in the way that epilepsy genetic testing has happened. And that's had a number of consequences for those of us who are involved in clinical testing and diagnosis and counseling. The first of which is that informed consent has become much more difficult. Instead of sitting down with a family and saying, based on your child's clinical features, we think it's very likely that she has a mutation in the PCDH19 gene, and maybe that's happening when the child is 2 or 3 years old. Now, instead, we have babies who are 2 or 3 months old who perhaps are admitted in the hospital because their seizures have been very difficult to control, and we are attempting to consent those parents for a test where we could get back any number of different diagnoses. And so it's neither practical nor desirable, I think, to sit down with a family and talk about 30 or 40 or 50 different genetic diagnoses when much of the time you might not get back a positive result, or you really can't predict what kind of result you might get back. So the end result is that I think often parents don't understand as much as I would like them to about what the test could mean for them in terms of the information that they're going to get back. And the other thing is, I think many of the clinicians who order the tests, because we don't see all of these families in epilepsy genetics clinic, and many of them don't see a genetic counselor, many of them really don't go through that informed consent process. And so the test is done, and they aren't actually even really aware that it's been done until they get results back. So I think there's a lot of misinformation sometimes with the parents as far as what they can expect to learn from the test in terms of a diagnosis. I think there is always an expectation that they will get a genetic diagnosis, which is not always the case. Many times these test results come back negative. And there's often an expectation, maybe because it's presented this way, that there will be treatment changes that can be made based on the genetic diagnosis. And in part, that's because these panel tests do include a small handful of genetic epilepsy diagnoses that are treatable. And I think sometimes the clinicians will sort of focus on those when they're consenting the families, and the families are going into the testing expecting that there will be some information about long-term prognosis, and there will be changes in medical management based on the test results. Another issue that's come up quite regularly for us now, in part because there's so many different labs doing these tests, and in part because the tests are relatively new, are that we get back many results that are of uncertain significance, or what we call VUS results, variants of uncertain significance, where the particular lab that's done the test has not seen that DNA sequence change before. It hasn't been reported in the medical literature. They think it's likely pathogenic based on the modeling and based on the fact that it's a highly conserved amino acid, but they can't say for certain that it is a real mutation, that in fact it is pathogenic. There's always the possibility that it could be a benign polymorphism that just happens to be in that family, and there's some other genetic explanation other than what's been reported. But we see those kinds of results very frequently. That becomes a bit of a problem when parents aren't available for follow-up testing. Usually when we get back a VUS result, the first thing that we would do is bring the parents in and get blood samples from them to see if one of them has the same variant that can help us interpret the finding. But if the biological parents either aren't available or won't come for testing, it can become that much more difficult to interpret a variant, and sometimes parents can become very upset. If it's a single parent, for example, I had one mom that I called back with a variant result, and she was much more upset than I would have expected her to be just at this news, and it turned out it was because the biological father was not available, would never be available for testing, and so she felt that this issue would never be resolved because he was never going to be tested. Another issue that comes up with the testing is that because there is this long time lag from the time that the sample is drawn to the time that the results come back, many of the parents have sort of forgotten that the test was done, or it's sort of so far in the back of their mind that when they get the call back from their clinician, they're taken by surprise if they aren't given advance warning that it's going to be a phone call about results. And I've had a number of families say things to me about the fact that they really weren't ready to get the results back, and that they wish their clinicians had sort of first contacted them to say, I'm going to be giving you your genetic test results back, and do it in person in the clinic so the family has a chance to be ready to hear that news, rather than having it, sort of unexpectedly over the phone 3 or 4 months after the sample was drawn. As well, I've had families tell me that sometimes clinicians will provide them with the diagnosis but not much information about it, and tell them to go home and look it up online, and that is obviously not helpful to families. So being prepared to give that information is extremely important. The other thing that I wanted to talk about a little bit before I go more into talking about our group of patients at Children's Hospital is what we've found with the parents in terms of just being ready to hear a genetic diagnosis. Some of the parents have been searching for an answer for many, many years. Their children are in their teens or their 20s, and they've been on what we call the diagnostic odyssey with many, many tests that have come back negative, and they're really hoping for an answer. And so you assume as the clinician that when you finally have an answer that this is going to be welcome news, but in fact it doesn't always turn out to be the case initially. I've seen many parents be somewhat overwhelmed by this news after so many years, and have told me that they really didn't actually expect ever to get an answer, even though they continued to search for one. So I think that's important to keep in mind. But on the other end of the spectrum, I think there are many parents of very young children, and I'm talking, just a few months old, who clearly have very difficult to treat epilepsy, but who have not shown yet any signs of developmental delay. It's too early to know. And for them, getting a genetic diagnosis very early from either a panel test or, as we will soon probably be moving to, in some instances, whole exome sequencing, I think sometimes those parents are really maybe not quite ready to have that information. And so I think it's very important, getting back to the informed consent, that when clinicians are ordering these tests, that they talk with the families about what potentially they're going to learn, and how ready they are to get the information, and in what context they want to learn that information, so the parents are emotionally prepared for that. Okay, so I don't speak Italian, and I don't write Italian, but I wanted to have one slide where I at least attempted to say something that those of you in the audience who don't speak English would be able to read. So these are photos of a few of the girls that we follow at Children's, and I want to say from one of them is, mom is sitting right in the room. All of these parents gave permission for me to show their daughter's photos and to talk a little bit about them. So just in broad terms, we are now following 8 girls with PCDH19 DNA sequence changes. 2 of them are siblings. And the age range is pretty broad, so not quite 3 years to 14 months. And the seizure onset mean has been about 7 months, with a range between 4 and 14 months. And I think just with one exception, all of the girls began having seizures in the setting of either fever or illness or immunization. This table just gives you a little bit of information about the phenotype that we've seen with our cohort and their corresponding genotype. Just by way of explaining some of these acronyms, DD stands for developmental delay, ASD, autism spectrum disorder, and ID, intellectual disabilities. So I think we have a somewhat homogeneous group of girls in our cohort as compared with what some of the other speakers have described, but I think that is in large part just because of the focus of our clinic and the fact that at Children's we tend to attract a younger group and a group of kids who maybe have tried to get diagnosis or treatment elsewhere, and where that treatment or diagnosis has not been successful. Now I want to make a comment about the genotypes, which is that only the ones in black were reported to us by the clinical labs as definitively pathogenic known mutations. The ones that are in green were all reported to us as variants of uncertain significance, although all of them, based on the modeling, are assumed to be pathogenic. This is what I was talking about before, the problem with VUS results. They are quite frequent in our experience, and in part because there are 6 or 7 or 8 different labs in the US doing this testing, and they don't all share data with one another, and there isn't necessarily a centralized database where all of this information is pooled. So sometimes I can get around that because all of these labs have genetic counselors, most of whom I know from my training, so I can call them up and say, oh, you know, this lab reported this result to me as a variant of uncertain significance. What can you tell me about your lab? But it's, you know, it's by no means easy to get that information. So in terms of inheritance for our cohort, the vast majority of these children, parental testing has in fact not been done, and I'll get to that in a few minutes. But there are 2 children for whom we know there's paternal inheritance. We know of 1 mutation that's de novo. The rest we don't know whether they're inherited or not inherited. So I wanted to give just a couple of case examples by way of explaining some of the counseling issues that we have encountered. So this is a little girl who we saw when she was not quite 3. I'm going to call her Fern. And she presented with refractory focal onset seizures at about 6 months following immunization. She proceeded to have clusters of partial complex and tonic-clonic seizures. Her issues developmentally tend to be more related to language. She has very poor articulation, so although she is quite social and interactive when you meet with her. It's very difficult to understand her speech. She has some gross motor delays and hypotonia and some aggressive behavior, but she has not been diagnosed with an autism spectrum disorder. Her issues tend more to do with oppositional defiant disorder. She has a variant of uncertain significance that's presumed to be pathogenic. And this is her family history. So you can see that there is some developmental issues and psychiatric history on both sides of the family. But we assume that probably the genetic change that's been identified in her came from her dad, because that's what has typically been seen. But we can by no means assume that, especially given the history on both sides of the family. Now, this family I've met in person. Mom, the parents are not together. The dad I've talked with at least 5 or 6 times to try to facilitate testing for him, and I have yet to meet him. He has not come to any of the appointments that I've made with him, and I've attempted to send a kit out to his house, but I think there are a number of reasons why he hasn't pursued testing. Part of them have to do with his own financial and social situation. He doesn't have health insurance. He doesn't currently have a home address. So he has not pursued testing. And I think Mom is somewhat frustrated because she's hoping to get an answer as to whether or not the genetic change in her daughter is inherited or not, because she has 2 other children, and I know she's particularly concerned about her older daughter, who has been diagnosed with a mood disorder and some speech and developmental delays herself, but does not have a seizure disorder. I think Mom is pretty convinced that her older daughter also has the PCDH19 variant. But as of now, we don't know that to be the case, although I think it's pretty likely at some point soon that we will test her. But it's been very difficult to counsel this family about the likelihood of recurrence, because we don't know for sure that this is in fact 100% a pathogenic mutation. And we don't know which side of the family it came from. If in fact it was inherited, it could be de novo. On top of that, this little girl also has a big stretch of loss of homozygosity on her X chromosome. So that could be a confounding factor as well. We haven't really known what to do with that piece of information. So sometimes when multiple genetic tests are ordered simultaneously, we get back several pieces of information that can make it very difficult to counsel about recurrence. The second girl that I would like to talk about is a bit older. She's now 14. I'm going to call her Amy. And she started having seizures at 14 months of age, but she was developmentally normal, according to the parents, until she was 3 years old, and apparently had complete developmental regression, loss of all skills, a few days following her measles, mumps, rubella vaccination. She has since had clusters of prolonged generalized tonic-clonic seizures, many episodes of status. She has a diagnosis of an autism spectrum disorder and a history of developmental delay. I know her behavioral symptoms have gotten significant enough that the family is considering placement outside the home. And she has both a known pathogenic mutation, which is the first one in black, and also a variant of uncertain significance, which we assume is probably a benign polymorphism, but again, we don't know that to be the case for sure. And this is the family history in this instance. There is an older brother who has some learning disabilities that I gather are somewhat mild, but he is on what we call an individualized education plan in the States and school. And then Amy's younger sister, who's now, I believe, 8, the parents have been holding off on vaccinating because of this history of the loss of skills and the developmental regression after vaccination. And I know that the parents are very much interested in testing. the daughter to find out whether she has a PCDH19 mutation, despite the fact that she doesn't have any symptoms of having a PCDH-related disorder. So that is something that might happen, although we as yet do not know the parent status in terms of the PCDH19 mutation. PhDs, and they really don't know anything about cells or genes or chromosomes, and I think that is still often the case, even for people of my generation in the States, that education around science is woefully inadequate unless that happens to be your major field of study. So trying to explain to somebody why something is genetic but isn't inherited can be a difficult task. And trying to explain for those individuals who do have some memory of learning about genetics, maybe they remember about the different inheritance patterns, it's very confusing to them why something that's X-linked doesn't follow the traditional pattern of inheritance from females to males that you see with things like Duchenne muscular dystrophy or hemophilia. So trying to sort of unlearn that information and introduce new information about the inheritance pattern can be quite difficult. Other counseling challenges for us, I talked about interpreting variants of uncertain significance. If parents are left with uncertainty about that, even after parental testing has been done, and living with that uncertainty and not knowing whether the diagnosis is in fact the underlying reason for their child's epilepsy syndrome, can be quite emotionally difficult. Some of the laboratories will voluntarily report back to us whenever they reclassify a variant. So a year later or 2 years later, if they determine that a variant is in fact benign or truly pathogenic, they will issue a revised report and contact the physician who ordered the test. But not all of the laboratories do that. So we can't always count on that information coming back to us without us asking for it. So sometimes it's up to the families to remember to come back to us every year and remind us, you know, hey, we need to check on this again and see if there's new information. And until they have that information, if you're talking about a young family that maybe wants to have more children and they're waiting on that information to make those decisions or to be able to have prenatal diagnosis, this makes it very difficult. Guilt and shame and blame, I talked about a little bit with the family that I mentioned that I saw as a student. But by way of explaining why so many of our parents haven't had parental testing, there have been a number of parents who have decided that they, in fact, do not want to have the parental testing done to determine whether the mutation or variant is inherited or not, because they, in fact, do not want to know whether it came from dad or whether it came from mom. So they refuse the testing for that reason. There has been at least one family that pursued the parental testing because they knew that it would help us to interpret the results of the variant of uncertain significance, but they asked us quite pointedly not to disclose the results to them. They did not want to know whether it came from mom or dad. And I think that's in part because in lots of families, there's a lot of finger pointing that goes on. Sometimes, you know, even before genetic test results come back, grandparents, for example, might decide that this, that, or the other thing came from that side of the family. And that can be very hard for marital relationships and family relationships. Some of our families choose not to share their child's genetic diagnosis with other family members, with teachers, with caregivers, because they're afraid it's going to bias the way those individuals interact with their child. So all of those things are things that we talk about during the counseling session before and after. Lastly, I just want to talk about resources for families. How many? I'm almost done. Huh? Yes, okay. I'm just about done. So unfortunately, I would say resources are rather scant in general for most of the genetic epilepsy syndrome. So at least in the US, when a child is diagnosed with something common like Down syndrome, there's these wonderful booklets that you can give to families called So Your Baby Has Down Syndrome. And they're beautifully written, unbiased information that's provided to families at the time of diagnosis, and we really don't have anything like that for most of the epilepsy syndromes, and certainly not for PCDH19, so that the families are often going to the internet, and sometimes the information they get is very helpful. Sometimes it may not be as helpful. One of the moms told me she never reads anything on the internet, and she never talks with any other families about her child's diagnosis, even families whose children have the same diagnosis, because she finds the stories about the other children depressing, and it makes her worry that her child will develop, the most severe end of the spectrum of symptoms. So she does not want to look at any of that information. I think in particular for that group, something that came from a medical professional might be more helpful if there were a balanced approach. So I would really put a plug in for the groups that are collaborating in terms of helping families to try to get those kinds of materials developed. I know many of the organizations have been started by families themselves. I had a few parents who told me that the primary reason they pursued genetic testing for their child was because they felt, even if it wouldn't change medical management, treatment, that it would give them a cause, and they have gone off and started these amazing support organizations, as one mom in the audience has done, and we are really quite excited to be working with all of the different groups that are involved in providing resources and funding to researchers. Just in terms of future directions, we have a research protocol that we're going to be starting comparing girls with PCDH19 mutations and those with SCN1A mutations, looking at the frequency of hypotonia as opposed to ataxia, spasticity, and gait abnormalities, because in our group we have found that hypotonia is more common in the girls with PCDH19 mutation and the others in girls with SCN1A mutations. I'm also going to be looking more systematically at counseling issues. And then we are talking with collaborators in the US about starting a PCDH19 specific registry, but I gather there are other international groups also talking about registries, so it would be nice to sit down and have a conversation about that. And then I just want to acknowledge our clinical and research team. Dr. Paduri is the only one in the white coat up here. And then finally, I just wanted to end by thanking our sponsors who fund our research and our clinical work, and then just invite any questions that you might have.